Hi and welcome back to my YouTube channel. In this video we will discuss about the parts of plant used. Resin used 2%, stem used 2%, latex 1%, leaves used 31%, aerial parts used 11%, fruit used 12%, bark 11%, flower 6%, seed 6%, and branches 3%. Active and herbal constituents. Acids properties are sure effect as antiseptic, cleansing, source citrus species. The second one is alkaloids, bitter, alkaline, nitrogenous compound, affects addictive affects central nervous system, toxic. The family source is Povera. The third one is anthraquinones, bitter, irritative, latexative, or source is rhum. Bitters properties bitter, affect appetizer and improve digestion. Fifth one is coumarins, smell of new mohe, affect antibiotic, anticoagulant. Family is Meltanus. Sixth one is flavonous, bitter or so sweet, often diuretic, antiseptic, antiplasmodic, anti-inflammatory. Their source is fugacum. The seventh one is glucosides. Properties is bitter, arid, uses as a antispasmodic, carcinogenic, sedative, effective heart rate and respiratory antibiotic, and the digestis lamina. Active herbal constituents is in which aids is gums and mucinous bland, sticky or slimming, smooth and soften. Resin acidic and astringent, antiseptic, healing. Sponin is sweet, stimulate hormonal, often anti-inflammatory or diuretic spawning in water. Tannins use as a antiseptic, checking bleeding and discharging. Volatile oils, aromatic antiseptic, fungal detal, irritate and stimulator. Steps involve the phytochemical screening. Random approach, toxic approach, phytochemical, ethanolic approach is information, plant materials, then extract, plant extract, then separation, pure compounds, then synthesize, structural elucidation and toxicology, structure modification. And steps involve the screening of herbs. In silica virtual screening, in silica molecular dynamic structure, in vitro biochemical and cell analysis, in vivo animal assays. New drugs. Experiential phases, exploratory study, experimental studies. Documentation of Rohart's clinical observed tolerance, interaction, dose range in patient of evaluation target activity in vitro and in vivo test. Biological screening, isolation and identification of active constituents, preclinical studies, toxicology studies, clinical trial, new drug. Indian traditional herb identification of the potential species, new herbal extraction fraction, new molecular structure modification. The second one is herbal formation, standardization, quality control, herbal formulation, preclinical study, toxicological study, and clinical trial. Research approaches to the herbal products. Field use standardized ethnomedicines, Ayurvedic use whole formulation, Ayurvedic therapeutics standardized, reverse pharmacological experiment documented exploratory studies, experimental research, clinical phase 2 and